Hi, my name is Claire Smathers, and today I'm going to be talking to you about bacterial group 2 introns. So, I'm assuming all of you know what bacteria are. Very small, sometimes unicellular organisms. And these bacteria have something called introns. Actually, we all have introns. So, what is an intron? So, whenever you think of a DNA strand and the genes that are on it, you think of those genes being converted to some sort of protein or having some sort of effect on the cell. So whenever these genes are actually transcribed and made into a protein, they're transcribed by the RNA polymerase, and the RNA polymerase goes along and transcribes this whole part of the gene, this whole gene, and converts it to an intermediary called an mRNA. And that mRNA is then converted to the protein that then can go and do the job. So what then is an intron? Well, these genes have two different parts. One part is the intron and one part is the exon. The exon is what we normally think of a gene being, the DNA that actually gives rise to a protein or some sort of function. But there's also these introns, and these introns are different because they are usually not actually transcribed and made into protein. So what normally happens is your gene is transcribed and you get this intermediary mRNA product, and the mRNA is consisting of both the intron and the exon. And what then happens is you get these intron parts that are cut out of the mRNA, and you get the exons put back together. And this new mRNA, called the mature RNA, is then what is made into the protein. So why am I talking about these introns when they don't really seem to have a use? Well, people used to think that introns were just what were called junk DNA. So we used to think that there were lots of our DNA that didn't really have a purpose, because we knew we had so much DNA but only so much protein, so what was the rest of the DNA doing? Well, contrary to that belief, introns are actually super important and play a lot of different roles. And I'm not going to get into all of the different things that introns can do, but what I am going to talk to you about is a very interesting particular class of introns found in some species of bacteria. And these are called the group 2 introns. There's also group 1 introns, but we're not going to talk about those. So what's special about these introns, and why are they different? So what's really interesting about these introns is they can actually act as a molecular machine. And some of you may have never heard that term before. So what a molecular machine is, it's a molecule such as a protein or DNA or RNA that can actually do a job. So it can do something like catalyze a reaction such as an enzyme does. And so these introns are actually what's called ribozymes, or RNA enzymes. So they will actually act to catalyze a reaction. So these introns can catalyze a reaction, which is really cool because we used to think that introns were worthless and didn't do anything. And now we know that this type of intron can actually do a specific job. So what do these introns actually do? Well, these introns are in their DNA, and they're actually in the RNA. So these bacteria have an RNA genome, and what the intron can do is it can actually remove itself from that RNA. So instead of what I talked about earlier, where you transcribed your genome into mRNA and then you remove the introns by a process of splicing, these introns are actually going to remove themselves. So they're going to splice themselves out of the actual RNA, and then they can actually move to other parts of the genome, which is even more interesting. So these introns can remove themselves and then hop around the bacterial genome. So I know this has been just a really short introduction into what bacterial group 2 introns are. But if you'd like to learn more, you can visit the Robart Lab website, or you can always come and talk to me after this talk. Thank you.